This protocol offers a quick and efficient strategy to simultaneously use two assays to screen several hundred compounds for their effects on cell growth. The assays can provide us information of whether specific compounds are cytotoxic, inhibit cell growth, or enhance cell growth. By utilizing the two assays together, we are able to increase accuracy and identify cytotoxic compounds. In cases where the results of the two assays diverge, this strategy may allow us to identify compounds with unique effects on cellular function that can be further examined by other means. The main advantage of this strategy is that it is quick, inexpensive, and non-labor intensive. Once mastered, it allows researchers to easily perform a primary screen of up to 200 compounds in 1 week, and then perform an exhaustive dose-response curve on 10 to 15 compounds in another week. Demonstrating the procedure will be myself and Rohini Manickam, a staff scientist and post-baccalaureate fellow from the laboratory.
First, in a biological safety cabinet, culture cells in a T75 flask with NSC medium so that they will be at least 80% confluent when beginning the assay. And place the flask in a cell culture incubator set at 37 degrees Celsius and 5% carbon dioxide. Once the cells reach 80% confluence, remove the flask from the incubator and aspirate off NSC medium. Add 3 milliliters of cell dissociation reagent and incubate for 5 minutes in the incubator. After incubation, add 7 milliliters of NSC medium in the flask and pipette vigorously to ensure all cells become detached. Transfer the dissociated cell solution to a 15 milliliter tube and centrifuge at 200 times G for 5 minutes.